Electron transport chains can be in chloroplast mitochondria and in prokaryotes. As a result of that, ATP synthase is going to generate ATP. When we talk about cellular respiration, we're going to get into glycolysis, the Krebs cycle, and the electron transport chain. If there is not oxygen available, fermentation will allow us to do repetitive glycolysis. So these electron transport chains, we have them in chloroplasts. So when we have these different electron transport chains, idea is that they're going to create a proton gradient, but we have different terminal electron acceptors. In chloroplasts, NADP plus becomes NADPH as it accepts the final electrons. In prokaryotes or bacteria, they also have electron transport chains and generate ATP through ATP synthase, but they don't have mitochondria. So they can occupy a lot of different regions and environments in the world because of the variety of terminal electron acceptors that exist in bacteria. So you can have nitrates, you can even have carbon dioxide to make methane, and you can have sulfates that often exist in like the deep sea vents. But we're going to focus on the electron transport chain within mitochondria. So as we get into that, we're talking about these terminal electron acceptors. And in this, we again have these redox reaction where we have oxidation and reduction. And oxidation is losing electrons and reduction is gaining electrons. So as we look at this, as we break down the carbon molecules, we're going to get NAD plus and FAD being electron acceptors, which are going to carry those electrons to the electron transport chain, where ultimately oxygen will be the terminal electron acceptor and water will be created. When we look at these formulas, the overall formula we're hitting with cellular respiration within mitochondria is that glucose and oxygen are going to produce carbon dioxide and water, and we're going to generate for the purpose of generating usable energy in the form of ATP. So we start out with glycolysis. Glycolysis is going to happen in the cytosol. It's not going to occur within the mitochondria. With glycolysis, we're going to make a little ATP. We're going to split the sugar, and we're going to carry some electrons. In the Krebs cycle, there is a preparatory reaction here, but the Krebs cycle is going to fully break down the carbon molecules to carbon dioxide. We're going to get a little ATP, and we're going to carry a lot of electrons. Finally, all of those electrons that are being carried are going to be used in the electron transport chain and chemiosmosis to generate the vast majority of the ATP we talk about in cellular respiration. So glycolysis, it has many steps. There's energy investment steps, there's energy harvesting steps, there's some phosphorylation that occurs. Ultimately, we start with glucose and we end up with 2 pyruvates. We put a little ATP in, we get more ATP out, and we carry a few electrons. So again, we have glucose, NAD plus coming in. We have to put a little bit of ATP in to begin with, but we get out our 3-carbon molecule pyruvate. We carry some electrons over to the electron transport chain within the mitochondria, and we get some ATP out of that. Now, before we can get to the citric acid or Krebs cycle, there is this preparatory reaction where the pyruvate gets transformed into acetyl-CoA. That process does generate a few electrons that are carried to the electron transport chain. But our next major step that we're going to focus on is the Krebs cycle. Now we are in the mitochondrial matrix, and we have this outer membrane of the mitochondria, and we have this inner convoluted membrane which increases the surface area. So we can have a lot more electrons being transferred and a lot more ATP synthases embedded. So in this Krebs cycle, though, again, there's lots of these intermediate steps which can get very confusing. Ultimately, the big idea here is that as the acetyl-CoA is coming in, we are ultimately going to get out the carbon dioxide. as a byproduct. We're going to make a little bit of ATP here, but the big idea of the Krebs cycle is this is where we generate a lot of electron carriers. We've got NADs and FADs. This FADH2 will get involved in the electron transport chain as well. So again, we've got our NADs and FADs coming in. We've got our acetyl groups coming in. And ultimately, we got to put in ADP and free phosphate in order to get ATP out. But the vast majority of what we're getting out of the Krebs cycle is focusing on these electron carriers. So finally, we get all of these electron carriers transporting electrons to the electron transport chain. As we look at that, here we have our NADH and our FADH2, and they are dropping off electrons. And these high energy electrons are powering these pumps. And we have this inner membrane of the mitochondria. We have the outer membrane that you can't see here of the mitochondria. We have this intermembrane space, and this is where the proton gradient is being created into the intermembrane space. So electrons power the pumps. These protons, or hydrogen ions, get pumped into the intermembrane space. ATP is made as these go through chemiosmosis. So the protons are diffusing back through ATP synthase, which then creates ATP. And this is where we get the vast majority of our ATP. It doesn't stay in the matrix. Ultimately, it's used by the cell for lots of different processes, but it's not quite done because oxygen needs to clean up. So all of these electrons that have been dropped off in the electron transport chain, oxygen needs to pick them up off of this final pump, and water is going to be created. If oxygen does not clean up, this backlog of electrons is going to stay on the electron transport chain, and NADH and FADH2 are not going to be able to drop off any more electrons. If they can't keep on dropping off electrons, we are not going to continue to pump hydrogen ions, and we're going to lose this proton gradient, which is then going to stop creating ATP. If we stop creating ATP with our mitochondria, that can be very dangerous. So fermentation is an alternative metabolic pathway if oxygen is not present. The big idea here is that those NADH that are heading to the electron transport chain and can't dump them off need to be recycled. You have to have empty trucks, empty NADs, in order to continue doing glycolysis and generating ATP here. So these electrons have to go somewhere because they can't go to the electron transport chain. NADH drops them off to create either a lactate or alcohol, depending on if it's in animals and bacteria or plants and yeast. So these byproducts ultimately become electron acceptors so that we can recycle the NAD plus and continue doing repetitive glycolysis to continue getting ATP out of carbon molecules like glucose. So in summary, we've got glycolysis, we get a little ATP, we create a little bit of electrons to be carried. This is happening in the cytoplasm. When we get into the mitochondria, that pyruvate is converted to acetyl-CoA, which gets into the Krebs cycle. We generate a little ATP here. We get carbon dioxide out. We get a lot of electrons being carried to the electron transport chain. The electron transport chain and ATP synthase is responsible for creating the vast majority of ATP through cellular respiration. So again, prokaryotes, chloroplast, mitochondria all have electron transport chains that generate ATP through ATP synthase. When we start looking within mitochondria, it's glycolysis, Krebs cycle, and electron transport chain that creates the vast majority of ATP. Fermentation can do glycolysis repetitively in the absence of oxygen so that ATP can still be created.